Then Linda will introduce our speaker. Then Paul will give the talk about the COVID-19 vaccine update. Then there will be the question and answer sessions. So his talk will be 30 minutes, and there will be the question and answer section 15 minutes. Then the Kshitij Karki from program director will closing the session. So this is our today's agenda. You can send the message if you have questions in the chat box, or you can raise the hand, and we will happy to receive any questions from your end. So we are still waiting for Linda. Linda is the senior investigator, and he's the medical anthropology based on my knowledge, and she's helping us a lot in our Nepal antimicrobial resistant program.
Well, thank you everyone for joining today's call. We have Dr. Paul Kilgore here, who I have worked with for many years, worked with him at the International Vaccine Institute. I'm sure it's going to be a very informative presentation. I know Paul has worked since the beginning of COVID, on vaccines. He was a principal investigator on the vaccine trials for Johnson & Johnson and Moderna in Detroit, Michigan in the United States. And welcome, Paul. And Paul has been to Nepal, and I'm sure he's hoping to see everyone face to face again in the future. But welcome to Paul, and thank you for giving the presentation this afternoon.
Thank you very much, Linda, Dr. Kalji. Thank you to all the GTA team as well, and Deepak, Karki, everyone. Really appreciate your help in getting me here today. So normally I would like to be able to do this face-to-face with you in Nepal, but this is also a good way to do it because I can share, I think, more information with you and give you really up-to-date information. So let me jump right into it. So one of the things that we know is that the pandemic continues. It's continuing globally. And one of the things that you can see here very clearly from this graphic is that in all regions of the world, we are seeing a continued transmission of the virus. And there's a couple countries where we even have some concern. Now, Europe is in a current state where they are actually seeing a rise in cases. They're seeing more transmission of virus. And so because of that, several European countries have entered into a new phase of lockdowns. This is quite new. In fact, France announced this past Friday that they're going to really lock down the area around Paris and other regions. Poland is doing a large lockdown for the next several weeks. And then when we look at other countries, we see Italy also is imposing another lockdown. Hungary, Bulgaria, Bosnia are tightening up restrictions. And now even in Germany, we're starting to see a rise in infections that is very concerning. And what it does appear to be happening is that we're starting to see the emergence and spread of these variant strains of the virus. And so we're going to talk about that today. The virus that we're really looking at is the structure that you see here. And it's the spike protein that is really one of the major proteins that we think about with coronavirus. The interior part, of course, the RNA, is the genomic mapping, and those are all the key parts that drive the virus replication. We also know that as people develop infections and antibodies to coronavirus, they make antibodies to several proteins, including the spike protein and the nucleocapsid protein. Now, the spike protein has this trimeric structure. It's three proteins that are interlinked, but there's one part of the protein that's very important. It's called the receptor binding domain. This part of the protein is also the part that is a place where antibodies will bind. So if you get vaccinated, for example, you'll make antibodies that bind to the receptor binding domain. But at the same time, the virus is very clever and it's developed mutations in this region, and that's changing the way that the virus behaves in humans. The way that we've thought about developing vaccines is actually kind of based on many years of vaccine development. So there's a lot of traditional ways to develop vaccines, the inactivated approach we'll talk about. We'll talk about the nucleic acid approach, the mRNAs. We'll talk about the viral vectors like the Janssen and AstraZeneca and other manufacturers. This is the adenovirus vector technique, for example. And then we have the subunit or protein-based approach. That's one that Novavax is actually working on. And you'll hear more about that from GSK and Sanofi as well in coming weeks. So if you look at this map or kind of depiction of the different approaches for vaccination, one of the things that you realize is that there's manufacturers all over the world that are working on these vaccines, which is great news. One of the things that's so important when you develop a vaccine for any pathogen is that you take multiple approaches, because back a year ago, we didn't know which of these approaches would work best. So it's really amazing that the global investment in the billions of dollars that went into developing these vaccines really was a global effort. There was some cooperation for sure. For example, the AstraZeneca is a cooperation between Serum Institute of India and Oxford University. And then there's other collaborations, of course. Pfizer and BioNTech in Germany was a really key collaboration as well in the development of different vaccines. And I'll come back to these in just a few minutes. But one thing I wanted to talk about is the way that we develop vaccines has changed over the years in a good way. And what I mean by that is that if you look on the left-hand side of this screen, you can see the orange is really kind of basic research, the discovery, the identification of the structure of a pathogen. So many, many years ago, back in 2003 and 4, when we had the first SARS pandemic that originated in China and spread to other countries, we knew that the structure of the coronavirus contained a spike protein. So that knowledge that we gained almost 20 years ago, we could apply in the case of COVID-19 to accelerate vaccine development. So that's one of the reasons why the phases of vaccine development could be accelerated. We could do it faster because we knew about the virus structure already. You know, when we develop these vaccines, we go through phase 1, phase 2, phase 3 studies. These have been done for all of the vaccines that we're talking about today. And then in the United States and similarly in other countries, the National Regulatory Authority, like the FDA, will go through a comprehensive review of all the preclinical data and the clinical data to make sure that, number one, the vaccine is safe. That's the number one goal. Number two, that it's effective, that it can prevent disease. And really, another thing that we analyze in that is the immune response. Does the vaccine generate a good immune response that is durable, will last for weeks, months, and even years when people get vaccinated? In the United States, we have one more review that happens at CDC, the Centers for Disease Control in Atlanta. That is done by the ACIP. The ACIP stands for the Advisory Committee on Immunization Practices. And that's an independent committee, separate from the FDA, separate from the CDC leadership, actually. And they make a recommendation on how to use a vaccine that gets approved by the FDA. And now for some of the vaccines, Pfizer, Moderna, and others, we are in the phase 4 period. And I'll tell you about that and what it means for us going forward. So if you look at the top screen here, normally we take about 15 years, maybe 20 years to develop a vaccine. But in the case of COVID-19 vaccines, we have the advantage of a couple key things. Number one, we knew that we had to develop a vaccine urgently. So all of the investigators got together and said, let's write the protocol for phase 1, phase 2, and phase 3 right now. So back in February and March of 2020, when the pathogen was just recently identified, people began to develop and write down the clinical trial protocols that would enroll human participants in the trial. Normally we phase it out, we space it out over a number of years. So we do, in the old days, we did one trial, analyze the data, report it out, get more money for the next phase, write the protocol, get the ethical approval, collect the data, analyze the data, make a report, and then go on to the next one, get funding for phase 3, and continue on that same process. Back last year, the US government, BARDA in particular, which is the Biomedical Advanced Research and Development Agency, committed billions of dollars to develop vaccines. So there was money up front, and that was a game changer in terms of vaccine development. If we had not had that money up front to develop the vaccines, we would have had to go down the slow road. But because the money was available, they could write the protocols, get the ethical approvals, and then get the enrollment going. Another big thing that you should know about these vaccine trials is that we do not collect data using paper anymore. We really collect the data using electronics, computers, smartphone apps, and we analyze the data very quickly because it's all electronic from the very beginning. It means that we have clean data, cleaner data than in the previous method, which used paper, and we can analyze the data much faster, get the reports out, and then get the data to the approval people and the regulatory authorities. So that's how we were able to speed up vaccine development. The mRNA approach is a unique approach. It's new, but it's really not that new. We've been working on DNA and mRNA vaccines for really 20, 25 years. The mRNA vaccines were developed for influenza, human metapneumovirus, human cytomegalovirus before COVID-19. In fact, Moderna was working on those other vaccines before they turned their attention to COVID-19. So that's Moderna and Pfizer's the kind of first type of vaccine. The recombinant SARS coronavirus 2 surface protein vaccines, those are the vaccines like GSK, Sanofi are working on, as well as Novavax and a couple other companies. Third approach, the viral vector approach, that's the AstraZeneca approach. That's the Janssen or Johnson & Johnson vaccine approach. And a couple others like the Sputnik vaccine from Russia are using the adenovirus to package the viral DNA so you can make the protein and then establish antibodies to that. One of the things I wanted to point out before I forget is this yellow line down here. When you get vaccinated, when you vaccinate others, the beauty of these vaccines is that you stimulate antibodies and you get T cell populations established that will have memory for the pathogen. So down the road, months or even years down the road, your immune system will remember the coronavirus and be able to mount a boosted immune response because of those memory B cells and T cells. I won't go into a lot of detail here on this table, but I'll make sure you have these slides because one of the things that's very interesting about this whole ecosystem of vaccine development is that it's very, very diverse. But one of the things that we are doing is using a lot of tried and true methods that we know work for vaccine development. For example, the adjuvants, the contents that are in these vaccines are things that we've used in other vaccines. So I feel very confident that when we go to make an RNA-based vaccine, for example, that those constituents, those things that you see here on the right-hand side, these are not things that just came out of the blue. They're not that new. These are things we've used before in other drugs and other vaccines. So that's good news. The mRNA approach is really, really amazing because we are actually taking up the mRNA in a lipid nanoparticle, basically a vehicle that brings the mRNA into your muscle cell. Once the mRNA is established in your muscle cell, it just borrows the machinery of your cell, the ribosomes, for example, the endoplasmic reticulum, the Golgi apparatus, to make the spike protein. Now keep in mind, when you get the mRNA, the Pfizer vaccine and the Moderna vaccine, the only protein that you're making is the spike protein. And that's the beauty of these vaccines. It's very important to have what I like to call a clean vaccine. It's making only one antigen, one protein, and that's what your immune system recognizes to create the antibodies, particularly the IgG antibody. Now, I wanted to mention the other approach from J&J. This is a good example of a adenovirus vectored vaccine, but we see that also with CanSino, the Gamaleya group from Russia, AstraZeneca. They're all DNA viral vaccines based on a adenovirus platform. In the case of J&J or Janssen, that's an adenovirus type 26. That's a human adenovirus that we've actually used and proven that it can be developed as a vaccine. We've used it for drugs in the past to deliver DNA, to deliver a gene for a specific disease. In the case of COVID-19, it's working. In fact, we've shown in the clinical trials with one dose, we have overall 85% efficacy against severe disease, 72% efficacy in US volunteers, but it's a global trial. So one of the things we know is that we have evidence that the vaccine works globally in South Africa, US, and other places. The Johnson & Johnson vaccine trial, this is our group at the hospital, at Henry Ford Hospital Health System. We have a large group. This is not actually everyone, but we have one site in a large global trial. It's called Ensemble 1, enrolled totally 45,000 people worldwide. And we were, out of many sites, we were in the top 20. We had more than 400 actually enrolled, including a very diverse group, including minorities, populations, which is very important to make sure we have good representation for this vaccine. Pfizer and Moderna are both recommended now, as you may know, in the United States and other countries. Similarly, AstraZeneca has been recommended, approved for use in Europe and many other countries now. Johnson & Johnson is going down that same road. Johnson & Johnson is now approved in the US. The Novavax vaccine, I expect to see an approval for Novavax in the next month or two, maybe earlier, because we know from the data in the European trials in UK, this vaccine is effective, about 89% efficacy at least. And the good news is that there's some efficacy against the South African variant we'll talk about. The Moderna data was very compelling. In a large group of people, about 30,000 people, it was 94% efficacious. The local adverse events with the vaccine, and now I'm talking about the first injection for the Moderna vaccine. On the left-hand side, you see pain. So local discomfort in your shoulder is what the main thing that people experienced. When we talk about the second injection, we also talk about discomfort at the injection site. That's the #1 local adverse event that people experienced. But you know, that's what we see with other vaccines, to be honest. It's been no different, and that's actually reassuring. What that means to me is that the vaccine is starting to induce a good immune response when people get vaccinated. Systemically, we see things like fatigue, headache, muscle aches, joint aches, but still the most common really were fatigue and headache, followed by myalgia and arthralgia. The others became much less common. And one of the things you notice here is that these were low-grade adverse events, grade 1 and 2 for the most part. Grade 3 and 4 are the more severe ones that we get really more attention to. But you can see very, very low-grade adverse events for the most part, which is encouraging. Now, for the Moderna vaccine, I just show you here a breakdown to demonstrate for you that the efficacy in both younger and older people is clearly there. You can see the point estimate in younger people is about 96%, in older people, 86%. So it doesn't matter the age group, we can see that the vaccines are preventing disease, including severe disease leading to death, hospitalization, and intensive care unit visits. Now, for the Pfizer vaccine, it looks quite similar. Overall efficacy, you can see 95%. But then when you break it down by age group, younger age group, 95% again, and then older people, 55 and older, 94%. So the bottom line with Pfizer vaccine as well is that even despite age group or breaking down by age group, the vaccine is effective in preventing death, hospitalizations, ICU visits, mild, moderate, severe disease. So it's very, very encouraging, actually. The 2-dose regimen, and note Pfizer and Moderna, both 2 doses, about a month apart. So Pfizer is 21 days apart. Moderna is 28 days apart, 2 doses. Very important, if you do get a 2-dose vaccine, no matter what manufacturer, very important to get both doses. The adenovirus approach is really, again, not new, but I just wanted to point out that we're basically taking out genetic information that's needed for replication. So these adenovirus vaccines do not replicate. It is impossible for these vaccines to cause disease, to cause COVID-19. That's true for actually the mRNA vaccines. It's true for AstraZeneca. It's true for Janssen. They cannot cause COVID-19. Impossible. I will say categorically impossible for them to cause disease because they cannot replicate and cause infection. They simply are exposing your cell and bringing in the new DNA genetic information to form the spike protein. The AstraZeneca results, again here, this is from the Lancet. AstraZeneca overall, 62%. The design of this trial was slightly different than the Janssen trial, different from the Moderna, different from the Pfizer vaccine. So if you're asked, why are these vaccine efficacy estimates different between these trials? One key thing to tell people is that the design of the trials was different. These were not identically run trials. Each manufacturer had a slightly different case definition, outcome definitions, laboratory testing was slightly different. The enrollment process was slightly different. The number of doses in some cases was different, like Janssen is 1 dose. So it's very important that people know that the design of the trials was different. That's one of the reasons why we see a difference in efficacy. Another key reason why we see different efficacy estimates is that the populations where these trials were run were different. So we know, for example, Janssen and a couple other trials had populations in South Africa. That can alter the vaccine efficacy if the local strain that is circulating is different from the original strain on which the spike protein gene is really derived, or from which the spike protein is derived for the vaccine. We'll get back to that in just a minute. AstraZeneca vaccine, this is the AstraZeneca local reactions, pain and tenderness. Pain and tenderness is really typical for any vaccine, but we're seeing it with the COVID-19 vaccines. And so that's not surprising at all. The systemic reactions, here also you see things like fatigue, very common. You see muscle aches, just very similar to what I just mentioned for Moderna and Pfizer, right? Also, we see headache, some malaise, just not feeling great for a couple days. And then a few more people will get fever and chills and other signs. But overall, these patterns that you see for the adverse events in all these vaccines is actually quite similar. The J&J vaccine I mentioned to you before, single dose. We are doing a 2-dose trial now for this vaccine. So that's just something to keep in mind, that you will look for results for the 2-dose trial for Johnson & Johnson coming up soon. But this vaccine at its core is really similar to AstraZeneca vaccine, actually. And the good news is that these vaccines can be stored at refrigerator temperature. I wanted to mention briefly the inactivated vaccine approach, because from Sinovac, we have CoronaVac, we have another one from Sinopharm. These do have an advantage, like the AstraZeneca vaccine, they can be stored at refrigerator temperature. They don't need a deep freezer like the Pfizer vaccine currently does. There is some data from late-stage trials in different countries for Sinovac and the Sinopharm vaccine. The Sinovac trial data has been published in some peer-reviewed journals. I'm still looking for the Sinopharm data, but they did a trial in the United Arab Emirates that showed about 86% efficacy for that vaccine. So these are inactivated vaccines. And if you look at this slide here, part C, I'll just focus your attention here. Basically, they use a vero cell culture. And the vero cell is very, very good at growing viruses. So if you infect a cell culture, vero cell culture with coronavirus, it will grow in the vero cells, and then you can extract that grown up virus, and then you inactivate it chemically and with heat. There's a couple other methods we can use to inactivate these vaccines, and then they're really constructed into a vaccine after that with some preservatives and other constituents. So that's the Chinese approach for a couple of vaccines there. Now, I would just want to mention briefly the mutant strains because I know that these are on top of mind for a lot of people. The UK strain is probably the most widely spread strain that is the variant right now, but we also have the South African variant, the B1351, and then we have a P1 variant from Brazil. These strains have mutations in the spike protein. It's the genetic code of the spike protein that's being altered to actually improve binding affinity. The B117, the UK strain, has been around now for several months. It is better at transmitting, so better at moving from one person to another, but also there's a report from the UK that suggests that this strain can cause more severe disease. The jury's still out. We have this strain now beginning to widely spread in the United States. It's all over Europe. So I would expect this strain to really emerge as the dominant strain in many, many countries. We have the South African strain. This was identified last year as well, but it's now spread to many countries. It's been in Asia, it's been in North America, it's in Europe now. So I expect this strain to also spread. The Brazil strain, the P1 strain, is very interesting because it actually caused infection, or I will call it reinfection, of populations that had been sick and infected with the original COVID-19 strain. Those people recovered, and then this new P1 emerged and then started to cause infections widespread in some locations. It does not seem to be spreading as quickly, but I do expect this one to continue moving from country to country. The new vaccines that we just talked about do work against these strains, but they appear to be less reactive. In other words, the antibodies that you make with some of these vaccines appear not to work as well for some of these variants. The South African variant seems to be the one so far for which these vaccines do not work so well. They do work a little bit, but not great. That's one of the reasons why the manufacturers are now working on developing new vaccines already for these new variants. Now, just last week, a study was published, and one of the things that this study in the Lancet showed is that the AstraZeneca vaccine was evaluated in South Africa, and they looked at the efficacy against the South African variant, 1351. Just read the bottom line conclusion. I won't be able to read all of this to you, but basically the 2-dose regimen for the AstraZeneca vaccine did not show protection against mild to moderate COVID-19 due to the South African variant. What does that mean? It means, number one, that we should still use the vaccine. It does not mean that we should not use the vaccine. The reason I say that is that depending on where you are, it could be Nepal or India, wherever you are, the predominant strains are likely not to be the South African strain right now. So you can prevent disease due to the other strains by getting the vaccine that we currently have. Okay, so I think that's an important message to get across. Do not throw out, we say don't throw out the baby with the bathwater. In other words, use the vaccine we have, and then later on we'll have a booster available that contains the spike protein for the South African strain. Vaccine deployment is never easy. It doesn't matter what country you're working in. We know that we want to get the vaccines out to as many people as possible. We want to do it in a fair and equitable way. Healthcare workers are typically going to be prioritized because they're at the front lines, but other essential workers are going to be key to helping restore economic viability in many countries. It's one of the reasons why the COVAX mechanism is being used. WHO and Gavi are working hard with that. Manufacturers are making agreements. But here, one of the things you see is that we really are still kind of in phase one. I'm hoping that many countries will get into phase two in coming months. But what that means is that we're going to have limited supplies at first. And so there's going to have to some prioritization of groups in the country. Hopefully those groups will take it up. There's one thing I could also communicate to you today. It is that when people are eligible to get vaccinated, encourage them strongly to get vaccinated. You know, in Nepal, of course, we have Covishield, the COVID-19 vaccine from University of Oxford with Serum Institute and AstraZeneca. And that vaccine is now rolling out. There's millions of doses now coming into the countries, including Nepal. But we still have challenges that we're going to be facing. One of the things is how to communicate to patients how these vaccines work, helping them navigate what choices to make, educating them about the complications of disease, weighing the risks and benefits. Are these adverse events, the local side effects, systemic side effects, are they worth it to prevent COVID-19? The short answer I will give you is absolutely yes, because one of the things that we see continuously all the time is that people who get COVID-19 just don't get COVID-19 and then recover and they go on with their lives. No, they actually suffer long-term effects in many cases after getting COVID-19. Brain fog, brain difficulties, headache, heart problems, other organ dysfunction, chronic fatigue, inability to go back to work, inability to carry on their daily activities appears to be a major problem with COVID-19. So we're very worried about that. That's being studied right now very carefully. I think the future is one where we're going to need to have more and more collaboration working together in Nepal with public health authorities, clinicians, hospitals, the community to improve prevention of COVID-19. We're going to need to have very, very strong surveillance. We need to detect these strains that are emerging early and as often as possible. And so we need to build up genomic epidemiologic surveillance in Nepal and other countries to make sure that we identify the spread and these new strains that emerge. And so finally, in the collaboration in Detroit, a lot is going on. We have testing, we have outbreak investigations, we have vaccinations, we have mobile units going out now into communities. So things are looking quite good, but it's a massive effort. It's going to continue. It's worldwide. And I think you're going to see a lot of great work going on in the next few months to develop the vaccines, get them out to the populations that need them. So with that, I think I'll stop and see if we have any questions. So thank you.
Thank you, Paul. As Paul said, we'll open up for questions now.
I have a question, Linda, to people.
Okay. Go ahead.
Does anyone have any reservation or any concern about receiving a COVID-19 vaccine?
Dr. Anup Bastola, maybe can you respond to the Paul's questions, Dr. Anup?
Or you can write it. You can write question in the chat.
Thank you, sir. Actually, like my experience, like Nepal, when vaccine came from India, there was a lot of rumors, miscommunication, and many of the healthcare workers they were reluctant to use COVID-19 vaccine, the Covishield from India. Okay. So many of my colleagues and friends, they said me, okay, we will wait for a few days to see the response. Okay. And later they accepted it. My director, and our team in Kilgore Hospital, we received COVID-19 vaccine. At that time, many of our colleagues in our hospital too do not wanted to take vaccine in early days. Later, they accepted it, and I think after the first phase of vaccination, when we started for frontliner other than healthcare worker, they also accepted very nicely. And very interesting fact about like elderly population, second phase of vaccination, most of the elderly population, they are very much willing to get this vaccine. That was so encouraging for healthcare provider like us. And in second phase, like it was a good number of elderly population, they received the vaccine. Thank you.
You know, that's a great point, Anup. And, you know, one of the things I wanted to mention to people is that for many years, I've known people at Serum Institute of India. As many people know, the Serum Institute of India is one of the largest, if not the largest, manufacturer of vaccines in the world. I mean, #1 in the world. They are a major vaccine supplier for UNICEF. So they're a trusted partner with WHO. That's the first point. Second is that I have visited firsthand their facilities in Pune, India. I've been on site there at their manufacturing plants. I've seen the care, I've seen the production that they have there. It is state of the art. It's top notch, it's world class. Any country would be very proud to have that kind of manufacturing capacity, and the dedication of the team there is amazing. Many people may know or recall that Serum Institute of India partnered with the FDA and the Gates Foundation many years ago to produce a new meningococcal vaccine for Africa. It was the only manufacturer to do that. So they made this serogroup A vaccine for Africa. And that vaccine is actually still being manufactured and used to this day. A very important example to show you the dedication, but also the care that they put into making vaccines. The other thing that's very interesting, I think, is the data that has been gathered in the trials for the AstraZeneca vaccine. And when I look at the adverse events for this vaccine, I look at the efficacy data, I will tell you personally that I would feel extremely comfortable recommending the AstraZeneca, the Covishield vaccine from Serum Institute of India to my family members, my father, my parent, my brothers, sisters, anyone in my family. I would recommend any one of these vaccines. It could be AstraZeneca, Moderna, Pfizer, Johnson & Johnson. Based on the data I've seen, the people I know running the trials, the places I know where the vaccine is manufactured, I feel very, very confident that these vaccines are well made, well developed, safe, and effective. So I would say no barrier to actually taking the vaccine. And it is encouraging that, as you mentioned, that the older adults are recognizing this vaccine is critical, and I would say critical to their survival, to be very, very honest. So I'm glad to hear that. Thank you.
Dr. Paul, I have some queries. I think all the participants, they are having the queries. Okay, there was an article in preprint in Lancet. Okay, so most of us in Nepal, because of the supply of COVID-19 vaccine, we might not get the second dose of this vaccine. So many of the colleagues, they have queries on like how the first dose, a single dose, can help. Second thing is like previously it was 4 to 6 weeks for second dose of Covishield, right? Now there are data showing like even after 12 weeks, it works very nicely. So the information that you have, can you share that? And second question is like, hopefully in coming days we will get Sinovac and Covaxin from India, okay? Because they have approved it in Nepal. So those are the kill vaccine, okay? So many of the colleagues, they were asking me, okay, the efficacy of those kill vaccine compared to adenovirus vector vaccine and other vaccine. Can you please clarify that? Thank you so much.
Sure. So for the one dose strategy with the adenovirus vaccine, so there's a couple of things to know. One is that, the Johnson & Johnson vaccine trial planned from the very beginning to be one dose only. And one of the things that they found in that trial with the design they had, it's a global trial, is that the vaccine was highly effective in preventing severe disease, hospitalizations, ICU visit, death, and moderate disease as well across the world. And I showed that data earlier. So overall, about 85% efficacy for one dose. Early on, when the UK started to roll out the AstraZeneca vaccine, they had the strategy of the one dose approach so they could maximize coverage in the population with one dose. They were criticized, and I think many remember that discussion in the global media news. So when they got that criticism, they started down the pathway to create the analysis and look at the efficacy of one dose and also the strategy of spacing out the dose, as you pointed out. And one of the interesting things that they found was that when they did space out the AstraZeneca vaccine doses to 12 weeks, they actually saw very good efficacy. So what that means is that in the original trial, they had a closer spacing. It was a matter of a few weeks. What it means now is that we can safely space out vaccine doses for the AstraZeneca vaccine and still end up with long-term protection, good two-dose efficacy, and you have a lot of people protected with one dose. It's my guess that if you get one dose of the AstraZeneca vaccine, you will be experiencing vaccine efficacy on the order of 50 to 75%. That's a ballpark range. That's an estimate. That's true. But you will have some protection with one dose, no doubt about it. That second dose that you may get 12 weeks later, approximately, that's important to get when it becomes available. So I would not delay that once you have access and you get ability to actually sign up for that. So do get the two doses ultimately. But the spacing out does seem to be working. The data I've seen in the preprint you mentioned, I think is looking good. The inactivated vaccines that you mentioned, it does appear that they are effective and efficacious, although the clinical trial data is not as maybe robust or strong that I've seen in all the vaccine candidates. One of the vaccine candidates did have trial results in the Lancet, so that did look good and effective. They look to be within the range of the other vaccines, perhaps a little bit lower, but not dramatically lower. So when you think about the efficacy of these vaccines, they do seem to work. I will tell you, though, the one thing we don't know about the inactivated vaccines versus the other vaccines or all these other approaches is the long-term protection. I mentioned earlier in my slides that we do know that with the Moderna vaccine, Pfizer, Johnson & Johnson vaccine, probably AstraZeneca as well, we do seem to have B-cell populations and T-cell populations that will help you establish immune memory. Long-term protection would follow from that. I don't know if we've seen the same data yet for the inactivated vaccines. So that's the one question I really have right now with the CanSino or Sinovac or Sinopharm vaccines. I really want to see long-term protection. So after people in the trial get vaccinated, I want to see antibody levels, T-cell subsets, 3 months, 6 months, 9 months, 12 months after that vaccine series to be reassured that those vaccines give long-term protection. They may need a booster ultimately. We just don't know right now.
Thank you. Thank you so much.
Thank you.
Oh, there's another question. Do you see it on chat about adverse events?
Yeah.
I do. I'm sorry. Let me read it to you. Some countries banned the COVID vaccine due to adverse events following immunization. In Nepal, some cases died, but we are unaware of the cause of death. So That's a great point, Dr. Joshi. So this came up last week, actually, the week before in Europe with the AstraZeneca vaccine, where some of the participants or some of the recipients of the vaccine experienced a blood clot. And so the question was, does the vaccine cause blood clots? The conclusion from the European Medicines Agency is that the vaccine is not associated with the formation of blood clots in those individuals. The other thing I need to mention to you is that when we started rolling out the vaccines for Pfizer and Moderna, this is worldwide, the early people that got vaccinated included healthcare workers, but also elderly. And when I mean elderly, I mean older people like in their 80s and 90s. We know, of course, that older people are going to have other concomitant underlying medical conditions that are going to put them at risk for serious disease and even death. It would not be unusual at all when you start vaccinating millions and millions of people, perhaps billions, that you're going to see people who get the vaccine and then die. It does not mean, though, that the vaccine is causing the death. They often, and typically what we've seen so far, is that the death, or in some cases the serious disease like a blood clot, is coincident with administration and receipt of the vaccine. When they go to investigate the clinical association and they look at the timing, so they look at the timing of onset of the disease, when the person got vaccinated, and what is the timing and the other underlying conditions that the patient has to learn if the vaccine is actually associated with that condition. There's also laboratory tests that are run. So there may be some immunologic tests or assays that are done. Other studies are done to do a really full investigation. So I think it's, the media will pick up, of course, on these conditions, these serious outcomes associated that appear to be linked to a vaccine. But in fact, in most cases, it appears to be that the vaccine is not linked to these conditions. And in the case of death, these deaths that are occurring typically are occurring in older individuals or individuals who have other serious underlying conditions that put them at risk for death to begin with. So I hope that makes sense. Every country needs AEFI investigators and surveillance to really make sure that no matter what, everyone who has an AEFI that's serious is investigated very thoroughly in a very systematic fashion. I hope that helps.
Thank you. Any other questions? Again, you can put them in chat or, just speak out.
One thing I would also mention, as people are just thinking, is that if they have a question, they can send me an email as well, or through GTA staff, I'm sorry. You can also send me an email or through Dr. Kalji. That's my email, of course. And don't hesitate. Please send me an email. I'd love to get your questions. And if it's not a question today, if it's a question next week, that's great too. I'd love to hear your thoughts.
Linda, I think there's no questions, so can you close the session, Linda?
Okay. So thank you everyone for joining us today. Especially thank you, Dr. Paul Kilgore, this was really an informative talk, and also very up to date. So it's nice to hear, what the latest information is that's available about these vaccines, and about the COVID epidemic. If there are no other questions, we will close the session. And as Dr. Kilgore said, if you have questions, you can send them directly to him or through Deepak or Karki at GTA. So thank you, and everyone have a nice Sunday evening. And thank you, and of course, thank you, Deepak and Karki and everyone else at GTA. They've been working with us to put together more of these phone conferences and Zoom calls because we've been unable to come and do some of the face-to-face programs that we've done. So thank you very much, Karki and Deepak, for setting this up.
Yeah, and I agree. Thank you very much, Linda. Thank you, everyone. And have a great evening.
Thank you.
Bye.
Thank you, Paul.
Thank you.
Thank you, Linda.
Thank you, Deepak.
Bye-bye.
Okay. Have a good day. Bye.
Bye.
Bye.